Microbiology is the scientific field that studies the smallest forms of life on our planet. By definition, microbiology is the study of microorganisms, living entities that are too small to be seen with the naked eye and require microscopes for observation. Microorganisms encompass several major groups. These include bacteria, which are single-celled prokaryotes, archaea, ancient microbes often found in extreme environments, fungi like yeasts and molds, protozoa, which are mobile single-celled organisms, algae, which perform photosynthesis, and viruses, which are cellular infectious agents. These microorganisms have diverse characteristics. They can be unicellular, multicellular, or acellular, range greatly in size, and possess remarkable metabolic diversity and reproductive capabilities. Microorganisms are ubiquitous, found virtually everywhere on Earth, in soil, water and air, on and inside plants and animals, and even in extreme environments like hot springs, deep sea vents, and polar ice. Their ecological and industrial roles are immense. Microorganisms drive nutrient cycling in ecosystems, decompose organic matter, contribute to food production, provide medical applications, and assist in environmental cleanup. Early predictions and discoveries about microorganisms span centuries of scientific investigation. The existence of microorganisms was predicted long before they were actually observed. Ancient scholars and philosophers like Aristotle speculated about invisible living entities that could cause disease. In 1665, Robert Hooke made the first recorded microscopic observation of microorganisms, documenting the fruiting bodies of molds in his book Micrographia. In 1677, Antony van Leeuwenhoek, a Dutch cloth merchant and scientist, became the first person to observe single-celled organisms using his handcrafted microscopes. Leeuwenhoek called these tiny living creatures animalcules and reported his findings to the Royal Society in London, documenting bacteria, protozoa, and other microorganisms. These pioneering observations laid the essential groundwork for the field of microbiology, revealing an entirely new world of microscopic life. The germ theory of disease was a revolutionary concept that transformed our understanding of illness and infection. During the second half of the 19th century, two pioneering scientists laid the foundation for modern microbiology. Louis Pasteur, a French chemist and microbiologist, made several groundbreaking discoveries that formed the cornerstone of the germ theory. His most famous experiment used specially designed swan neck flasks to disprove spontaneous generation, the incorrect belief that life could arise from non-living matter. Pasteur demonstrated that microorganisms were responsible for fermentation processes, and that these microbes always came from other pre-existing microbes. He also developed the pasteurization process, which uses heat to kill harmful bacteria in liquids like milk, preventing spoilage and disease. His swan neck flasks allowed air to enter, but trapped microorganisms in the curved neck, proving that microbes in the air, not spontaneous generation, caused growth in sterile broths. Robert Koch, a German physician and microbiologist, further developed the germ theory through his methodical research and groundbreaking techniques. In 1876, Koch conclusively demonstrated that Bacillus anthracis was the cause of anthrax disease, providing the first definitive proof that a specific microorganism causes a specific disease. He developed solid culture methods using gelatin and later agar, which allowed individual bacterial colonies to be isolated and studied. Koch went on to identify the bacteria responsible for tuberculosis in 1882 and cholera in 1883, further confirming the role of microorganisms in disease. Koch established a methodical set of criteria, now known as Koch's postulates, to prove the relationship between a specific microbe and a specific disease. First, the suspected microorganism must be present in all cases of the disease. Second, the microorganism must be isolated from the diseased host and grown in pure culture. Third, the cultured microorganism should cause the same disease when introduced into a healthy, susceptible host. Fourth, the microorganism must be re-isolated from the experimentally infected host and identified as the same original organism. The germ theory of disease revolutionized medicine and public health, marking a paradigm shift in our understanding of illness. It replaced the miasma theory, which incorrectly attributed disease to bad air or vapors, with the understanding that specific microorganisms cause specific diseases. This new understanding led to the development of antiseptic and aseptic practices in surgery and medicine, dramatically reducing patient mortality. Public health measures improved with new emphasis on sanitation and hygiene standards in urban planning, food production, and water treatment. The germ theory provided the foundation for the development of vaccines and later antibiotics, saving countless lives. Together, these advances dramatically reduced mortality from infectious diseases throughout the 20th century and continue to guide medical practice today. The work of Pasteur and Koch continues to influence modern medicine and microbiology, establishing a foundation that scientists build upon to this day. Ferdinand Kohn was a German biologist who laid the foundation for modern bacteriology in the 1870s. He was the first to classify bacteria as plants and established a systematic approach to bacterial classification based on their morphology and physiological characteristics. Kohn's classification system divided bacteria into four main groups based on their shapes: spherical, rod-shaped, filamentous, and spiral bacteria. Alexander Fleming made his groundbreaking discovery in 1928 when he returned from vacation to find that a mold had contaminated one of his bacterial cultures. He observed that the penicillium mold had created a bacteria-free zone around itself. This zone of inhibition showed that the mold was producing a substance that killed the Staphylococcus bacteria. Fleming's discovery of penicillin marked the beginning of the antibiotic era. For the first time, doctors had an effective weapon against bacterial infections. This breakthrough revolutionized medicine, saved countless lives, and opened the door for the development of many other antibiotics. It fundamentally changed how we treat infectious diseases. The work of pioneers like Kohn and Fleming laid the groundwork for modern microbiology and continues to influence research and medical practice today. Artificial intelligence and machine learning have become integral components of modern microbiology research, revolutionizing how scientists analyze microorganisms. AI systems enable researchers to analyze vast microbial datasets with unprecedented precision. These technologies identify complex patterns, accelerate discoveries, and significantly improve the accuracy of microorganism classification. Advanced genomics and metagenomics techniques have transformed our ability to study microbial communities. These approaches allow researchers to analyze genetic material directly from environmental samples, revealing unprecedented levels of microbial diversity. Microfluidics technology has emerged as a powerful tool in microbiology, allowing researchers to manipulate small quantities of fluids with unprecedented precision. This technology enhances single-cell analysis and enables high-throughput screening of microorganisms. Together, these advanced technologies, artificial intelligence, genomics, and microfluidics, are transforming microbiology research. Their integration is accelerating discoveries and revolutionizing our understanding of microbial life across multiple disciplines. Antimicrobial resistance represents one of the most significant threats to global health. Antimicrobial resistance occurs when microorganisms develop the ability to defeat the drugs designed to kill them. The scale of this problem is immense. By 2050, it's estimated that drug-resistant infections could cause 10 million deaths annually, surpassing cancer as a cause of death. Resistance develops through a straightforward but troubling process. When bacteria are exposed to antibiotics, resistant mutations can survive and multiply. Current research focuses on multiple strategies to address this crisis. These include discovering novel antimicrobial compounds, developing combination therapies, and targeting bacterial communication systems. Other promising approaches include bacteriophage therapy, which uses viruses that specifically target bacteria, and antimicrobial peptides derived from natural sources. Advanced diagnostic tools play a crucial role in combating resistance. Rapid molecular diagnostics and next-generation sequencing allow for quick identification of resistant strains, enabling more targeted treatment approaches. Point-of-care testing and AI-driven platforms are bringing sophisticated diagnostics directly to patients, reducing the time to effective treatment. Addressing antimicrobial resistance requires a coordinated global effort, combining innovative research, careful stewardship of existing drugs, and advanced diagnostics. Microbiology plays a crucial role in environmental sustainability through various applications. Environmental microbiologists study microorganisms in soil, water, and air ecosystems, where these tiny organisms have enormous impact. Bioremediation is a process where microorganisms clean up environmental pollutants. These specialized microbes can break down contaminants in soil and water. For example, certain bacteria can degrade oil spills, heavy metals, and even radioactive waste. This natural approach is more sustainable than chemical treatments. In waste management, microorganisms are essential for composting and breaking down organic materials. Bacteria and fungi efficiently decompose food scraps, plant materials, and paper waste, converting them into nutrient-rich compost that enhances soil fertility. Microorganisms are the backbone of nutrient cycling in ecosystems. They drive essential processes like carbon fixation, decomposition, nitrogen fixation, and mineralization. These processes maintain soil fertility, support plant growth, and help ecosystems remain balanced and productive. Without microbes, these natural cycles would collapse. Biofuels production is another crucial area where microorganisms contribute to sustainability. Various microbes can convert organic material into renewable energy sources. For example, yeast ferments sugars into ethanol, while certain bacteria can produce biodiesel. These microbial processes offer alternatives to fossil fuels, reducing our carbon footprint. The demand for environmental microbiologists is growing rapidly as we face increasing environmental challenges. Researchers are developing advanced bioremediation techniques, engineering microbes for plastic degradation, and creating microbial fuel cells for electricity generation. Other areas of focus include sustainable agriculture solutions and climate change mitigation strategies. These innovations all rely on understanding and harnessing the power of microorganisms. In summary, microorganisms are critical partners in building a more sustainable future. Their versatility and powerful metabolic capabilities make them indispensable tools for addressing our most pressing environmental challenges. The field of microbiology is expected to grow significantly in the coming decade. Projections show a 7% job growth for microbiologists from 2023 to 2033, which is faster than the average for all occupations. The financial outlook for microbiologists is positive, with a median annual wage of $87,330 as of May 2024. Success in the field of microbiology requires mastery of specific laboratory techniques. These include PCR, spectroscopy, microbial culture methods, DNA isolation, and microscopy. Equally important are professional skills like communication, statistical analysis, and scientific writing. The future of microbiology is increasingly interdisciplinary. Progress in the field requires collaboration across traditional departmental boundaries, including genomics, biochemistry, data science, medical sciences, environmental science, and biotechnology. As these interdisciplinary connections strengthen, microbiology will continue to drive innovation and scientific advancement across multiple disciplines.